Okay, now that we've solved now for the combined multiphysics of the diffusing species and for the velocity profile of the solvent, we have to do some post-processing. Because we had the transport of diluted species originally not solved for, it didn't automatically produce a graph. But that's easy enough to add. All we have to do is right-click over here on results, add a new 2D plot group. Now we have to select the data that we want to use in this 2D plot group. So we're going to right-click again on this 2D plot group. We're going to select a surface plot. Okay. And on this, within the surface plot, we have to specify what it is we want to solve. The default here is the velocity, which we don't want to do. What we want to solve for, replace the expression that's right here. We want to solve for the concentration. So if that's not immediately apparent, when you scroll down, you can go to transport of diluted species. You can select species C, and you have a tremendous number of options of things that you can plot. So the first thing we're going to do is plot the concentration. So once we have that selected, you can have other options here to specify how it looks on the screen. Let's zoom in to extents a little bit better. We just have to hit up here on this plot, which is the pen with the different colors coming off of it, and it should plot the concentration distribution on there. Let's zoom to extents again. And as you might expect, for a point source of a diffusing species into a laminar flow field, that it would fan out and have a distribution that looks like what you're seeing here. So let's grab this and move it around a little bit, if we can here. So let's go back to zoom extents. And let's specify the zoom in right here on this region. So this is near where the flow is coming in. It develops fully developed laminar flow. Dye is injected here and it's forming this distribution. Not too surprisingly, fans out as you go down the x-axis in this velocity distribution. There are many different plots and ways to get data from this, and I encourage you to explore these distributions. But we can add, if you right-click on these, you can add other types of plots over the color contours. For instance, let's add an arrow surface plot above this plot that's currently there. And we will have normalized arrow sizes, and you can see them on this distribution here. So the normalized arrow sizes are distributed like this, where they're all the constant. We don't want necessarily normalized, so let's do proportional. Now you can see this nice, what you'd expect for a laminar flow distribution velocity profile that is quadratic between those plates. You can change the scale of these arrows by sliding this slider bar. So these arrows remain proportionate to that particular flow pattern, but you can see what the velocity distribution looks like. There's a very large number of different things that you can plot. So let's play around a little bit and add another distribution, another 2D plot group. We can also do one-dimensional plots that extract particular pieces of data by slicing through the profiles of different regions. So let's right-click on this again, and then now let's look at a contour plot. Let's see, contour. What do we want to look at? Let's look at a contour plot of the concentration level sets. Then you can hit plot, and it should show the concentration level sets as this transport convection diffusion equation progresses from one end of this to the other. Let's zoom in. You can definitely refine the number of levels. You can refine and add extra data points, smooth out the lines. You can increase all these different variables for post-processing analysis and production. Let's see if there's a way here to improve the ability to interpolate along these elements. You can also view element properties in post-processing via another different type of plotting command. So really there's just a tremendous number of types of plots that you can make. So data set, you can have multiple runs and get different runs and plot them all at the same time if you change parameters. So once you have this model set up nicely, it becomes really easy to do parametric studies. You can do bona fide continuation studies as a built-in package and modify the study parameters here, but you can also just play around with it a little bit and change the properties of the fluid or the species that's diffusing. For instance, let's make this diffusion coefficient an order of magnitude less and see how that impacts the distribution. So we've made this change here. Let's go back to the study and solve this again. And then we can re-examine the distributions to get a qualitative understanding about how this has changed. Check the convergence here as it's running. It probably will take a little bit longer because diffusion has this effect of smoothing things over, and when that goes away, it typically takes longer to do a run. It becomes more of a stiff problem. Numerical stiffness is a problem when you're solving the nonlinear equations. Should be settling down to a solution here in a moment. There it goes. A little bit more time here, it's getting closer. This might actually fail to converge unless it starts to go lower error more dramatically now, which it still is not reaching that limit very fast, but it seems to have converged without an error. If there is a convergence error, it'll give you a big splash screen with an X through it. It says there was an error in the convergence, it didn't meet tolerance. So here's what the concentration distribution looks like now when we reduce the diffusion coefficient by an order of 1 order of magnitude. So it takes a lot longer down this distribution for this diffusion to dominate. The number that captures this ratio is the Peclet number, and it's the ratio of the diffusion time to the advection time in this type of domain. So this is just a rambling video, but there's a lot you can do, a lot you can look at after processing, and feel free to ask questions if you have particular things you're interested in looking at. Thank you.